So we are very pleased to welcome you to this new webinar. It is our first webinar under the new Timal Life Sciences banner. This webinar will be dedicated to skin inflammation and innate immunity. If we look deeply in the skin is a tertiary lymphoid organ. When an external stimulus stimulates the skin's immune system, for example, infectious antigens or allergens, first, we have an activation of the skin innate immune cells that are Langerhans cells, dendritic cells, macrophages, natural killer cells, neutrophils, and eosinophils. Then, there is an activation of adaptive immune cells such as cytotoxic CD8 T lymphocytes that kill infected or cancerous cells, but also CD4 helper T lymphocytes that create polymorphic inflammation. For example, Th1 in connectivitis, Th2 in atopy, and Th17 in psoriasis. And finally, but not represented here, B lymphocytes that give rise to the plasma cells and produce antibodies. So in the field of immune inflammation, you can first use the in silico virtual screening. If you have a molecule and you want to know which target it will bind to, or if you are a step further and you would like to modulate your molecule's effect toward finding the right candidate. Thanks to in silico screening, we can predict docking between a molecule and its targets and find application for it. We will not develop this today. We have already presented this solution during a previous webinar, so please ask if you wish more information. We offer to evaluate here the efficacy of your compound in vitro with isolated immune cells or fresh whole blood.
Fresh whole blood is a very simple but very informative assay format. We can test many types of compounds, such as biologics, small molecules, nanoparticles, proteins, peptides, or even devices. Readouts related to the global response or to specific cell types can be analyzed. Most of the time, these are cytokines and chemokines released in supernatants. For products that can activate an immune response, such as yeast or bacteria, blood is very sensitive to detect the presence of very low concentrations. The human whole blood we use has numerous advantages, as this is a fresh material coming from a strong partnership with the French blood bank. It contains all cell types and plasma, so it has all functional blood cells in their natural matrix with no additives, no antibiotics. Cells interact together within the matrix and with the matrix. So this provides a physiological relevant model complementary to the use of isolated cells or cell lines, but here with, let's say, untouched cells. Another benefit in using this assay format is that we can test at the same time tens of conditions on blood from different donors, thus analyzing inter-individual variations. For cosmetic applications, blood is mainly used as a reagent to give a quick answer about the safety. Indeed, in the case of screening of many ingredients, plant extracts, for example, blood is a powerful tool to eliminate candidates that trigger unwanted immune responses and allergenicity, and to focus on those that do not induce such responses, thus allowing to work on ingredients with the safest profile for further bioactivity characterization. Among the safety parameters we can test using fresh whole blood are the hemocompatibility and the detection of pyrogens. Hemocompatibility evaluation relates to several tests, such as hemolysis, which is the capacity to induce red blood cell lysis, or leukocyte activation. Both can be measured using fresh blood as a reagent. Pyrogens are fever-inducing contaminants, usually of microbial origin. They can be present, They can be present, for example, in plant extracts.